Welcome to this journey into the microscopic world of cell biology. Today, we are going to explore a tiny but absolutely critical structure inside our cells called the nucleolus. It might just look like a dark spot under a microscope, but it is the very foundation of life. By the end of this lesson, you will understand not just what it is biologically, but how we can mathematically measure its incredible productivity. To truly understand the nucleolus, let us imagine a massive, bustling city. In this analogy, the nucleus is the central city hall, holding all the master blueprints for everything the city needs to function. Inside this city hall, there is a highly active, highly specialized department whose only job is to build the construction workers of the city. In our cells, these construction workers are ribosomes, and the specialized department that builds them is the nucleolus. It is the origin point for all cellular construction. Now that we have our analogy, let us look at the actual biology. Formally, the nucleolus is a dense structure found right in the middle of the nucleoplasm of eukaryotic cells. Unlike many other cellular compartments, it does not have its own lipid bilayer membrane to separate it. Instead, it is a tightly packed, busy gathering of ribosomal RNA and proteins. Because these materials are concentrated so intensely in one place, it appears as a very dark, dense spot when viewed under an electron microscope. So, how does this dense spot actually form in the first place? It assembles around specific areas of our DNA known as nucleolar organizer regions, or NORs. These regions contain multiple repeating copies of ribosomal DNA genes that are actively being read and transcribed by the cell. Because it lacks a rigid membrane, the nucleolus is highly dynamic and constantly changing. It actually disassembles completely when the cell divides during mitosis and then carefully rebuilds itself once the cell division process is complete. Let us visualize this structure. On screen, you can see the entire nucleus surrounded by its porous envelope. Deep inside is the nucleolus. If we magnify it, we see it is not just a solid blob, but has a highly organized internal architecture. The process starts in the fibrillar center, moves outward through the dense fibrillar component, and finishes in the granular component. This outward flow represents the step-by-step assembly line of ribosomal RNA. Having seen its structure, we must ask, why does this specific region matter so much to our survival? The answer lies in its product. Every single protein in your body, from the keratin in your hair to the hemoglobin in your blood, is built by ribosomes. The nucleolus is the exclusive site where the RNA components of these ribosomes are transcribed and assembled. If this factory were to shut down, the cell would quickly run out of ribosomes, protein synthesis would stop, and the cell would die. This critical function brings us to an important medical application in the real world. Because the nucleolus is the primary engine of cell growth, doctors and pathologists pay very close attention to its size in patient biopsies. Abnormally large nucleoli are a classic, undeniable hallmark of cancer. Cancer cells divide rapidly and uncontrollably, which means they require massive amounts of ribosomes to fuel their continuous growth. A swollen nucleolus is a clear visual indicator of this dangerous, unchecked cellular hyperactivity. Now that we understand the biological and clinical importance of the nucleolus, let us look at it through a quantitative lens. To truly appreciate the sheer scale of work happening here, we must realize that a growing mammalian cell produces millions of ribosomes before it can divide. This requires a highly coordinated and rapid transcription process. By modeling this production mathematically, we can understand exactly how a cell scales up its manufacturing capacity when it needs to grow or respond to new demands. This brings us to our first mathematical model. The total rate of ribosome production, denoted by the variable R, is equal to N times K. Here, R is measured in ribosomes per minute. The variable N stands for the total number of active ribosomal DNA genes located inside the nucleolus. The variable K represents the transcription rate, which is how many transcripts a single gene produces per minute. If a cell needs more ribosomes, it must either activate more dormant genes to increase N or speed up the transcription process to increase K. Let us apply this formula with a concrete example. Imagine we are studying a rapidly dividing human cell. Through observation, we find the nucleolus has exactly 400 active genes. We also measure that each gene produces 25 transcripts per minute. We want to find the total production rate. We take our formula, R equals N times K, and substitute our known values. Multiplying 400 by 25 gives us 10,000. This means the nucleolus produces 10,000 ribosomes every single minute. Now, let us scale this up to see the long-term productivity. We just found that the nucleolus produces 10,000 ribosomes per minute. To find the total production over the course of 1 full hour, we must multiply this minute rate by our time factor. Since there are 60 minutes in an hour, we multiply 10,000 by 60. The final answer is 600,000 ribosomes per hour. This staggering number perfectly illustrates why the nucleolus is so dense and active. This incredible production rate requires physical space. The physical size of the nucleolus is directly proportional to the cell's demand for protein synthesis. A higher demand requires a larger factory. Because the nucleolus is roughly spherical, we can use standard geometry to calculate exactly how much the factory expands when a cell becomes cancerous. As we will see, even a small increase in the radius leads to a massive, exponential increase in total volume. To measure this physical expansion, we use the standard geometric formula for the volume of a sphere. The volume, V, equals 4/3 times pi times the radius cubed. In this equation, V represents the total volume, measured in cubic micrometers. Pi is our standard circular constant, approximately 3.14. The variable R represents the radius of the nucleolus. Notice carefully that the radius is cubed. This mathematical reality means that even a small increase in the radius will result in a massive, exponential increase in the total volume of the factory. Let us calculate the baseline volume for a healthy cell. Suppose a normal liver cell has a nucleolus with a radius of 1.5 micrometers. We plug this radius into our volume equation. First, we cube the value of 1.5, which gives us exactly 3.375. Multiplying this by 4/3 and pi yields a final volume of approximately 14.14 cubic micrometers. This is the baseline factory size required for normal cellular maintenance. Now, let us look at a cancerous liver cell, where the nucleolar radius has doubled to 3.0 micrometers. We plug this new radius into the exact same equation. Cubing 3 gives us 27, which is significantly larger than our previous multiplier. Multiplying by 4/3 and pi gives a final volume of approximately 113.10 cubic micrometers. Even though the radius only doubled, the actual manufacturing capacity increased by a factor of 8, supporting rapid tumor growth. We have covered a lot of ground today. To summarize, the nucleolus is the indispensable ribosome factory located right in the heart of the nucleus. It is a dynamic structure that scales its production mathematically based on the number of active genes and transcription rates. Furthermore, its physical volume increases exponentially to meet high cellular demands, which is why it is a key indicator in cancer diagnosis. You now have a comprehensive mastery of both the biology and the mathematics of the nucleolus.